Hi, everyone. Welcome to our Tuesday get-together on the cerebellum. I know many of you are busy with Society for Neuroscience posters, and I hope to see many of you in a week. So today, as our guest, we have Tim Murphy and Edward Yan. Tim received his PhD and did postdoctoral training here at Johns Hopkins University. He worked with Joe Coyle and Jay Baraban. And from his early work as a graduate student, he was interested in looking at stroke and recovery, and he made a major discovery regarding the relationship between glutamate and the neuronal cell death. Um, and since then, he has continued to work on neural mechanisms of recovery from stroke. And you may know that there's been recent work showing that cerebellum can play a role in the recovery from stroke. There was a recent study showing deep brain stimulation of the cerebellum in people is influential in recovery from stroke. We're also going to hear from Tim's graduate student, Edward Yan, who is a PhD student in Tim's lab. Edward graduated from McGill with a degree in pharmacology and neuroscience. Thank you both for being with us and look forward to your presentation.
Oh, thanks so much, Reza, and it's really a pleasure to kind of be back in Baltimore, at least on Zoom. So I'm just going to give a brief introduction and then turn it over to Edward, who has started this really interesting project on the cerebellum and then gotten us into really a new, for us, a new area and I think some new opportunities. So our lab has done a lot of wide field imaging lately, and sort of the perspective is looking down on the mouse, like we were kind of flying over it in a helicopter, and then looking down on both hemispheres, and then not using two photon, but just using single photon imaging to resolve regional kind of map-like activations. And you can see the cerebellum back here. And so what Edward has done is really helped us leverage some methods to look at relationships between spiking in cerebellar neurons and these larger cortical maps. And there's lots of ways in which you can look at these maps. Initially, we used voltage-sensitive dyes, like the Peterson lab and Grinwald labs. And then more recently, we use genetically encoded sensors. And one shortcut you can take is if you just want to look at these regional activations using genetically encoded probes like GCAM, you don't need to take the skull off. You can do a transcranial form of imaging. And that's what we do for genetically encoded indicators. Here's some broad scale maps. So this is a wild type mouse. You see these on the left, you see these kind of regional motifs. And this is work of Majid Mahajirani, who is now at McGill. And so wild type mouse. And then in an acallosal mouse, the two hemispheres are kind of asynchronous. The maps aren't as well developed. And so we're going to use a similar kind of methodology to look at relationships between the cerebellum and these big cortical maps. We also do some functional imaging during behavior, so reaching. There isn't any cerebellar imaging here, but this is something that we want to do, is go into sort of task-based imaging, and then also potentially mesoscale two-photon cellular imaging. So at the heart of this work that Edward's going to talk about is a kind of an event-triggered analysis of these kind of cerebellar cortical relationships. And we've been inspired by work of others. This is a nice review article by Logothetis on kind of event-triggered whole brain methods. And so we started doing an event-triggered imaging in the cortex, and this was with Dongsheng Shao. And he would record with a 16-channel neuronexus probe and would isolate spike trains and do kind of a spike-triggered averaging. Edward is going to take this one step further and do this with neuropixels probes in the cerebellum. So this is our analysis. So in these early studies with the neuronexus probes, we would do spike sorting, we'd isolate cortical or subcortical units, and then we would do some statistics and kind of get these cortical spike triggered maps. And so this is just a schematic of the approach. So we isolate these spikes, and then we would zero in on the cortical activity just before and after these spikes. And Edward is going to do something similar using correlation. And one of the advantages of this method is you can do this kind of event-triggered imaging almost anywhere in brain. Let's see. So here's some maps emerging. This is with this electrode was in the barrel cortex, 2 spikes, you don't really see much. If you go up to 32 spikes, you start seeing a specific pattern emerging, and this is across the entire cortex. And then here, 512 spikes. This is a kind of a typical barrel cortex motor map. And so this is a spike-triggered average map, and you can see as you get up into the hundreds of spikes, you can get a nice kind of crisp map. So when this neuron fired in barrel cortex, it was also associated with GCamp activity, let's say in this anterior motor area. You can do this, as I said, anywhere in the brain, and I don't want to take away from Edward's talk, but these are some barrel cortex maps. Here we put the electrode in the thalamus, a couple different thalamic nuclei. Here's the 16-channel neuronexus in the striatum in a different map. And so now Edward is going to do this in different regions of the cerebellum. Can even be done with peripheral neuron spiking. So this is facial motor nucleus, and this was a study that Edward and Dongsheng just published in eLife. So this is a kind of a facial motor nerve cortical GCamp triggered map. So this is the cortical GCamp activity associated with the timing of those motor nerve spikes. So I'm going to stop sharing now and turn it over to Edward, who's going to tell you about the application of similar approaches in the cerebellum.
All right. Okay. Can everyone see my screen okay?
Looks good.
Okay, so yeah, thanks for this great opportunity for me to present my work that I did in Tim's lab for the last couple years. The project is titled Decoding the State-Dependent Cortical-Cerebellar Functional Connectivity in the Mouse Brain. Yeah, so for the past couple of decades, there has been really a great evolution of how we view the function of the cerebellum. Traditionally, we consider the cerebellum to be primarily responsible for sensorimotor processing. This is shown by this classic pencil-style somatotopic map of the cerebellum. With the advancing neuroimaging techniques, this functional map of the cerebellum has been really refined with higher resolution. And also, it was discovered that the cerebellum is not only involved in sensory and motor tasks, it actually plays a role in a wide range of cognitive tasks as well. And this observation is extended to mouse models, where it was found that the cerebellum is important in cognitive functions such as reward prediction, social behavior, aggression, etc. And so a natural question that arises from this observation is how does the cerebellum regulate this wide range of tasks? So there are really two main layers of circuit complexity that we think that contribute to the computation capability of the cerebellum. So firstly, there exist non-uniform functional zones that are mostly organized in a sagittal way in the cerebellum, and these zones can be differentiated based on their gene expressions, cellular properties, plasticity mechanisms, and also their involvement in different learning tasks. And the second layer is the input and output pathways of the cerebral circuits. So a key piece of this puzzle is the communication pathways between the cortex and the cerebellum. So these two structures co-expanded throughout evolution, and they form this multi-synaptic closed loop connections where the cortical information flows first to the pontine nuclei, which then propagates to the cerebral cortex through the multifiber granule cell Purkinje cell pathway. The Purkinje cells synapse onto the DCN, which then synapse onto the thalamus, which finally comes back to the cortex. And we think that understanding the details of this closed loop connection will be really helpful for us to understand the physiological function of the cerebellum, because particularly in the mouse cortex, the cortex is very nicely topographically segregated into different regions, and we know a fair amount about what exactly do each of these cortical regions do. And so knowing like which cortical regions that the cerebellum is functionally connected to, this could potentially give us some information about the circuit organization and also the physiological functions of the cerebellum. So the focus of this talk will be on characterizing the functional connectivity between the cerebellum and the cortex. And there has been several state-of-the-art technologies developed to simultaneously monitor cortical and cerebellar activity. These techniques include, but not limited to simultaneous two-photon imaging, simultaneous electrophysiological recordings in the cortex and the cerebellum, and also a combination of miniscope calcium imaging in the cortex and electrophysiology in the cerebellum, which we heard a couple weeks ago. However, since our goal is to gain a more complete picture of which part of the cortex that the cerebral neurons are functionally connected with, we decided to leverage mesoscale cortical imaging, a technique, as Tim has introduced, that is capable of capturing neurodynamics of the entire dorsal cortex of the mouse brain. And we combined this with neuropixel recording in the cerebellum, which would allow us to characterize the functional association between different cortical regions and single cerebellar neuron dynamics. And here is an example of the spike trains along the neuropixel probe that span multiple cerebellar layers. So some more details about our experimental setup. So in order to capture state-dependent changes in cortical cerebellar functional connectivity, we performed the recording first when the animal is anesthetized using 1% isoflurane. And then following that, we perform recording also in the awake state. And throughout this recording session, the animal is lying on a plate without doing any specific tasks. And in total, we did about 120 recording sessions in 35 mice. And from the reconstruction of the neuropixel probes, you can see that we recorded from the medial cerebral cortex all the way to the lateral cerebral cortex, and also we're able to reach the deep cellular nuclei neurons. In order to get the functional connectivity patterns of individual cellular neurons, we computed the partial correlation between delta FOF of each pixel in the mesoscale cortical activity and the spike train of individual cerebellar neurons, and from which we can construct a partial correlation map, which really tells us which part of the cortical regions is most correlated or anti-correlated with single cerebellar neuron activity. We also shuffle the spike train to generate null correlation values in order to determine the statistical significance of these correlations, and we mask out the regions that are not significantly correlated. And in this particular example, we see that this cerebral neurons activity is positively correlated with primary sensory cortex cortical activity and anti-correlated with the retrosplenial cortical activity. So to see what the most common functional connectivity patterns are, we clustered all the correlation maps into 7 main groups. And the way we do this is that we first aligned each individual correlation map to the Allen brain atlas, and we computed the activity index, which is defined by the fraction of pixels in each cortical regions, as we can see here, that are significantly correlated with cerebellar activity. So for each correlation map, we can generate a activity index vector, which is then used to cluster all of these maps. And the way that we the reason why we chose to use activity index is because this reduces the impact of small patches of activation, and also allows us to take into account of the fraction of pixels that are not significantly correlated with the cerebellar activity. And so in total, we got about seven clusters of these connectivity patterns, and here it's organized from anterior dominated patterns all the way to posterior cortex dominated patterns. And what we can immediately appreciate from this is that not only other correlation maps that are dominated by high correlations in the motor cortex and the sensory cortex, there are also a significant number of cerebellar neurons that are primarily correlated with the retrosplenial cortex, the visual cortex, or a combination of both. And we can also see the individual activity index of these correlation maps arranged in anterior to posterior manner. This observation is also evident when we specifically looked at the overall distribution of the cluster assignment of cerebellar neurons that are in each of these cerebral cortex lobules or the deep cellular nuclei. What we found is that in each of our recorded regions, there existed neurons that had correlation maps that belong to each of the 7 clusters, indicating that these functional connectivity patterns with the cortex really diverse in the cerebellar regions that we recorded, and this is not limited by the anatomical boundaries. We also compared the distribution of these correlation maps between anesthetized state and the awake state, and we found is that there is a trend in increase in the number of neurons in the awake state that belong to cluster 3, which correspond to high correlations in the limb sensory cortex trunk, sensory cortex and the motor cortex. This sort of corresponds to the notion in the literature that these verbal anterior regions of the cerebellum is involved in limb actions. And it's possible that in the awake state, there are increased number of neurons that receive more information from these sensory modalities. But overall, as we can see from the left bar and the right bar, that there isn't too much of a change in the overall distribution of these cluster assignments between anesthetized and awake state on a population level. However, this picture quickly changed when we looked at the functional connectivity patterns on a single neuron level. So here we plotted the histograms of the similarity between correlation maps in anesthetized state and wake state of individual neurons. And as we can see, there is a wide distribution in each of the cerebral regions, which suggests to us that the individual neurons correlation maps can actually change quite dramatically in between the two states, which is in contrast to what we saw before, that on a population level, this overall distribution seems to be relatively stable in between the two states. So in order to verify...
May I ask a question?
Yeah.
Did you identify the neurons in the cerebellum?
Yeah, so we're gonna come to that in next few slides. Yeah. So to verify the this effect, this change in the correlation maps is really due to the change in the brain states of the animal. We in a subset of the mice, we continued anesthesia. We started another anesthesia reporting sessions after the awake sessions, and we compared the correlation maps between the two anesthetized state. And what we found is that the correlation maps is actually quite stable between these two interleaved anesthetized states, which is in contrast to what I showed here, which is that in between the anesthetized state and the awake state, the maps can be quite different. In order to eliminate the possibility that what we see here, the change in the maps are just due to movements in awake state, we computed the correlation maps using only periods where the mouse are not moving in the awake state, and compared that to the maps that were calculated from the full-length recordings. What we found is that removing the movement period from the recording doesn't really impact the overall patterns that we see here, suggesting that what we see here, this kind of state-dependent changes in the functional connectivity patterns is really due to the changes in the brain state of the animals. Next, we wanted to see if there is any cell type difference in terms of their functional connectivity patterns with respect to the cortex. And specifically, we identified putative mossy fibers and the Purkinje cells in our recordings, which corresponds to the input to the cerebral cortex and the output respectively. So we identified putative mossy fibers based on their triphasic waveforms and the narrow gap in their autocorrelograms. And we identified putative Purkinje cells based on their regular and high firing rate. And this part was done with the help from Phylum, which is a Phi plugin developed by Navarro itself. And when we looked at the distribution of the functional connectivity patterns of these two cell types, what we found is that, again, they both had diverse functional connectivity patterns with the cortex. In the Purkinje cells, we see a slightly higher fraction of neurons that showed posterior cortex dominated patterns. We also compared the distribution between anesthetized state and awake state. In the multi-fiber case, we see quite a significant reduction in the number of neurons that belong to cluster 2 in the awake state, but an increase in cluster 3, which could indicate an increase in kind of information from the sensory cortex instead of the motor cortex in the awake state for the multi-fibers. But in the Purkinje cells, overall on a population level, again, we don't see too much of a changes in the distribution of these maps. And consistent with what we saw before, on an individual neuron level, these maps can be quite fluid in between these two states. One very important feature of Purkinje cells is that they can integrate two streams of input, one from the parallel fibers and the other from the climbing fibers, which originate from the inferior aura. The influence of the parallel fibers inputs is manifested in changes in the simple spike firing rate, whereas the climbing fiber input can be directly measured by complex spike rate. And these two input pathways also have different origins in terms of the relationship to the cortex, where the cortical information flows through the pontine nuclei, which give rise to the parallel fiber input, whereas the climbing fiber input comes from the mesodiencephalic junctions. And so what we want to do here is really to see if these two inputs can have different cortical representations. So we decided to compare the functional connectivity patterns of simple spikes and complex spike pairs in the same Purkinje cells. And these pairs can be identified based on their cross-parallelograms. As we can see that there's a pause in the simple spike activity following a complex spikes. So in total, we found about 20 pairs of simple spikes and complex spikes of the same Purkinje cells. This is mainly limited by the fact that complex spikes fire a lot less, and very few complex spike chains have statistically significant correlational patterns with the cortex in our recording setup. But when we compared the cortical representation of the simple spikes and the complex spikes of the same Purkinje cells, we found that there is a spectrum of how similar they are. So they can be very similar in these cases in both the anesthetized state and the awake state, but also they can be quite distinct. However, what we noticed is that in most of the complex spikes cases, their functional connectivity patterns are predominantly correlated with the motor and somatosensory cortex. And this difference in the similarity between the complex spike and simple spike correlation maps really arises from whether the simple spike correlation maps are dominated by anterior or posterior cortex. And indeed, when we looked at the overall distribution of these correlation maps on a population level in complex spikes and simple spikes, we see that the complex spike distribution is biased toward the first three groups, which represent higher correlation with the sensory and motor cortex, whereas the simple spike connectivity patterns with the cortex is much more diverse. So just a brief summary for the first part of the talk. I showed you that we can observe diverse cortical connectivity patterns in all recorded lobules of the cerebellar cortex, as well as the DCN. And while these functional connectivity patterns of individual cerebellar neurons displayed marked changes across different brain states, the overall assignments to specific cortical areas at the population level remained stable. And finally, the complex spikes of Purkinje cells preferably associated with the sensorimotor cortex, whereas the small spike activity displayed more diverse cortical connectivity patterns. So next, we wanted to know whether there is any relationship between how the local, the cerebellar circuits is organized and their long-range functional connectivity pattern with the cortex. So firstly, one can imagine maybe it's possible that the neighboring neurons may share more similar functional connectivity patterns, but the neurons that are more far apart are more likely to show distinct maps. Therefore, we plotted the physical distances between each pair of the cerebellum neurons and the similarity between their functional connectivity patterns. And what we found is that there isn't really a clear relationship. Neighboring neurons can have both very similar and very different connectivity patterns, and at the same time, very far apart cerebellum neurons can also have either similar or dissimilar correlation maps. Well, so next we ask, if not the physical distance, is it possible that the local synaptic connection is a better indicator of how similar their long-range functional connectivity patterns are? So to determine whether a pair of neurons are synaptically connected, we adapted this method from this paper published by the Buzaki lab, where we took two spike trains and first computed their cross-parallelograms, and we specifically focused on this narrow window with lags of 1 millisecond to 4 milliseconds, which corresponds to monosynaptic connections. And what we want to see is whether there is a peak or a trough, which corresponds to increased firing probability or decreased fire probability following the spike of one of the neurons. And we also jitter one of the spike trains by plus and minus 5 milliseconds, and using this to generate a null threshold in order to determine a statistically significant peaks or troughs, which correspond to excitatory connections or inhibitory connections between pairs of cerebellum neurons. With this, we can compare the correlation maps between a pair of excitatory connected neurons or a pair of inhibitory connected neurons, as well as not connected neurons. And when we evaluated the cumulative distribution function of the similarity between the correlation maps of a pair of neurons, what we see is that in the excitatory pair group, this distribution is biased towards 1, which suggested that a pair of excitatory connected neurons are more likely to have similar functional connectivity patterns with the cortex. On the other hand, the inhibitory pair distribution is biased towards minus 1, suggesting that they're more likely to be dissimilar. However, this effect that we see in the inhibitory pair seems to go away in the awake state. So taking these results together, we think that this polarity of the synaptic connections, but not the physical distance between the cerebellum neurons, can bias the similarity between their connectivity patterns. And there is a subtle difference between the anesthetized state and the awake state. Seems that the inhibitory kind of functional modules within the local cerebral circuits are more dissociated in terms of their long-range relationship in the awake state. So the next question that we wanted to probe is whether the spontaneous functional connectivity patterns that I showed you before that are informative of the physiological functions of the cerebellar neurons. So it would be great if the spontaneous activity that we recorded can actually tell us something about the functions of these cerebellar neurons. So in order to study this, we included a sensory mapping period where we gave the mouse either facial stimulation or hind limb stimulation. And after the sensory mapping period, we proceeded with our normal anesthetized spontaneous recording, where we computed the functional connectivity patterns from. So first, we confirmed the cortical response to either the hind limb stimulation or the facial stimulation, and we showed that indeed it's the contralateral sensory cortex that corresponds to the modality, either the hind limb or the face stimulation, that showed the highest activation following the stimulation. And then from the sensory mapping period, we can identify cerebellar neurons that are responsive to hind limb stimulation or facial stimulation. So we know the functional identity of these cerebellar neurons. And then we compared the functional connectivity patterns of the group of neurons that respond to hind limb stimulation. And again, this functional connectivity is calculated during the spontaneous recording period, where the animal is not receiving any external stimuli. And what we see is that the hind limb responsive neurons do show a very unique average pattern in terms of their correlation with the cortex, and this they show a higher correlation in the motor cortex and the hind limb sensory cortex, and this is also evident when we looked at the activity index, where in the hind limb sensory cortex we see a significant bilateral increase in the activity index in the hind limb stimulation responsive cerebellar neurons. On the other hand, when we looked at neurons that respond to the facial stimulation, they also show a very specific connectivity patterns. And this is also different from the hind limb stimulation responsive neurons, where you see this higher correlation in the oral facial sensory cortex. And when we looked at the activity index, we see a significant contralateral increase in the oral facial cortex correlation. And so what this really tells us is that this functional connectivity patterns that we recorded during the resting state does have some biological meanings. It can tell us something about the physiological functions of the cerebellum neurons, at least in the sensory function framework. And this kind of effect is also modality specific, where the hind limb stimulation and facial stimulation would have their corresponding cortical regions have higher correlation with those cerebellar activities. So finally, given that we previously observed that the single cerebellar neurons can display a very dynamic functional connectivity pattern between anesthetized state and the wake state, we wondered whether that is actually a reflection of a change in the underlying sensory processing capability between the two states. So to test this, we first measured the hind limb responses of cerebellar neurons in both anesthetized state and the awake state. And then we recorded the 10 minutes spontaneous activity in both anesthetized and awake state, where we again calculated the correlation maps from. Indeed, we found two subsets of cerebellar neurons that responded differently in these two states. The first group only responded to hilum stimulation in the anesthetized state, but not in the awake state. And we call this the anesthetized only group. Whereas the second group only respond to hilum stimulation in the awake state, but not the anesthetized state. And if indeed the spontaneous cortical-cerebellar connectivity patterns are indicators of which sensory stimulation the cerebellar neurons respond to, we should expect the anesthetized only group to show increased correlation with the hilum sensory cortex in anesthetized state, but not awake state, and vice versa for the awake only group. And that's exactly what we found. So in the anesthetized state, we showed that they only had a increased correlation with the anesthetized with the hilum sensory cortex and the motor cortex in the anesthetized state, but not the awake state. This is also shown by the significant increase in the activity index in the anesthetized state compared to the awake state. In the awake-only group, we see the opposite effect. So in this group of neurons, only in the awake state, they showed a increased correlation with the hilum sensory cortex and motor cortex, but not in the anesthetized state. So this really shows that this kind of this state-dependent change in the sensory processing, specifically the hilum stimulation, is accompanied by a modality specific changes in association with the cortical areas. So to summarize the second part of the talk, I showed you that the local synaptic connections between the cerebellar neurons can bias their long-range connectivity patterns. And the spontaneous functional connectivity patterns between the cerebellum and the cortex aligned with individual cerebellar neurons' functional responses to external stimuli in a modality-specific manner. And the tuning properties of the subsets of the cerebellar neurons differed between the anesthetized state and the awake states, which is mirrored by state-dependent changes in their long-range functional connectivity patterns. So in addition to the two layers of heterogeneity in the cerebellar circuits that I showed you previously, we propose that there is an additional layer, which is the state-dependent adaptation of these functional connectivity between the cerebellum and the cortex that may give rise to flexibility in functional identities of these cerebellar neurons. And with that, I'd like to thank everyone in the Murphy Lab, especially Tim, who gave all the help along the way. Dongshan, who really pioneered a lot of these functional connectivity studies, and also mesoscale cortical imaging in the lab, as well as Purkinje for surgery assistance. Yeah, with that, I'd like to take questions.
Thank you so much, Edward. Let me start with a statistical question for you. So you showed that complex spikes activity was more correlated with sensorimotor cortex. Simple spikes were more diverse. But of course, complex spikes suppress simple spikes. So there's a statistical issue with regard to how can you separate these two inputs.
Yeah, sure. So I think what we are seeing here on in terms of the long-range kind of functional connectivity is on potentially a slightly slower time scale than the kind of like cross correlograms that we see here in terms of the when we identify the pairs of simple spike and complex spikes. So I think it's possible that even though the complex spikes do kind of inhibit on a very fast time scale the simple spike activity, on a more like slower time scale, there's still this kind of difference or similarities in their kind of functional connectivity patterns with the cortex.
Just other questions for Edward. I'll go with another one for you. I guess I was expecting you to show us maps of the cerebellum rather than only maps of the cortex. Maybe I missed it.
Yeah. So I guess so. So I guess the way that we showed sort of the distribution is through when we just group the cerebellum neurons into different anatomical regions, and we showed that like there is a diverse distribution in each of the regions. We did indeed try to map the kind of the identity of these cluster assignments back to the 3D reconstruction that I showed here. But because like these, I guess these probes are sort of like just along one dimension, and they're not really kind of super close to each other, we couldn't really identify a spatial patterns in terms of their kind of organization, in terms of their functional connectivity patterns. And so like it really sort of just kind of dispersed throughout the entire cerebellum, that there are kind of these all of these cortical patterns throughout the cerebellum.
May I ask you a question?
Yeah.
So I was wondering if you also looked at the mossy fiber functional connectivity, and if it did correlate with the simplest spike activity that you saw. Like, is there a mossy fiber complex spike comparison of the map, functional connectivity map that you've done, or?
So are you talking about like a mossy fiber?
Yes.
Yeah, so we looked at mossy fibers and Purkinje cells. We compared the overall kind of distribution of these connectivity patterns, and I believe in some of the so we couldn't, like, obviously the mossy fibers and the Purkinje cells aren't really monosynaptically connected, so we didn't identify them in this part. But overall, like in terms of the overall distribution, we did compare them.
So you expect them to be like the similar results as the simplest spikes? So the mossy fibers that are projecting would explain the simplest spikes that you're observing, and the differences that they have with the complex spikes?
Yeah, so I suppose I suppose the implication here is that the simple spikes are a manifestation of the parallel fiber pathways, which got its input from the mossy fibers, and this kind of comparison between the complex spikes and simple spikes kind of extends a little bit to that of the comparison between the simple spikes and the mossy fibers.
There's a question on the chat from Christopher Chen. He's asking if you have tried different anesthesia in your recordings.
Yeah, so no, unfortunately, we didn't try different anesthesia. We only used a light anesthesia, so 1% isoflurane.
So I'll follow that up. There's been a couple of papers that have it's been at the older papers that describe a biphasic activity of Purkinje cells under anesthesia that they did not see when the animal was awake. I wonder if you've seen any differences in the two states.
Yeah, so I think there is definitely differences in their activity between anesthetized state and wake state. We don't see a huge there is a wide distribution of changing firing rates. There is a reduction in some cases. There is a increase in some cases. In some of the cells, actually, we do see a very like slow oscillation in neuronal activity in the anesthetized state, but not the awake state. I don't know if you can see this here down here. You see this very regular kind of peaks of firing rate, and these oscillates on time scale of minutes, which is quite interesting. But we didn't really sort of follow up on this sort of. But I think it's not restricted to Purkinje cells. However, it's also observed in other cell types that we recorded from.
Wonderful. Any other questions for Edward and Tim? Edward, any questions that we should have asked you that we did not?
I don't know. I think this is sort of the main findings in our paper. I think I've included most parts. There are a couple controls. I guess I mentioned the firing rates changes didn't really impact the connectivity pattern changes that we saw in between the states. Yeah, I can think of it's tough to be a reviewer for yourself.
All right. Tim, Edward, thank you both.
Oh, thank you. Yeah, it's been a pleasure.
Bye-bye.
Bye.